All right. Hello, everyone. My name is Chris Metler. Um, this is my midterm presentation. It's called Trouble Upstream, the Parasite Problem in Sea Otters. And this is for OCB 4303 with Professor Jeremy Kiska. All right, so today we're going to talk about this in a few sections. It's going to be first introduction and background. Second, we're going to go over the sea otter. This is mainly going to be about the California sea otter, and the disease in particular that's plaguing them is it's called Toxoplasma Gondii. The timeline is probably the most important portion. That's the meat and potatoes of the presentation, and then we're going to go over a quick conclusion. All right, so the sea otter disease. Toxoplasmosis, a deadly parasitic disease, has in recent years been linked to the death of sea otters along the coast of California. And in recent years, I mean more like around the 90s, but it's still an issue. Excuse me. So just going over the California sea otter in general, scientific name is endangered. So it preys primarily on marine invertebrates, like sea urchins, what else? Marine snails, things like that. It's a tool user, by the way, so it uses rocks to crack those things open. Probably my new favorite animal. They're pretty cool. They live in kelp forest habitats, so temperate environments. They are a keystone species, and by that we mean if we were to eliminate the sea otter population in their ecosystem, the ecosystem would collapse or at least suffer catastrophic damage. Specifically, they're a keystone species because of the prey that they eat. They act as a control mechanism. So if they were to be eliminated and the sea urchins ran rampant, they would destroy the ecosystem. So in general, they fell from around 300,000 individuals in Northern America to around 1,000 to 2,000 a long time ago, just due to the hunting for their coats. They have the densest fur in the animal kingdom. It's very soft and very sought after, so people were just hunting them almost to extinction. The California population specifically fell to, I believe, around 50 individuals at one point, and now all of the sea otters in that area on the west coast stem from that 50 sea otter population. Currently, they're still listed as endangered, unfortunately. So, going over Toxoplasma Gondii, the disease itself, this was discovered in 1908 by Nicole and, I'm going to mess this up, Manchot in Tunisia. It's a parasitic disease, of course. It's an apicomplexan, which is a phylum of parasitic alveolates. Basically, all you need to know is it's a protist. It's not an animal or a plant. It just falls under eukaryote in a different category. So it can infect humans and many other warm-blooded animals. And actually, I learned this during this assignment, up to 50% of the world's population is actually infected with some strain of Toxoplasma Gondii. It's just most people are non-symptomatic, so we've just learned to live with it. I did not know that, and that kind of shocked me going over this. So it can cause coordinative and physical changes. Sometimes it tends to infect the brain, so sometimes it can cause issues that were never foreseen. New symptoms are still popping up around the world, so it's still hard to pin down sometimes if somebody is infected with Toxoplasma Gondii. There's also preliminary research showing that it can cause behavioral changes, which is pretty scary for a disease. So it's spread mainly through exposure to infected cat feces or undercooked food. And that you'll see during this presentation is one of the biggest issues, is specifically cat feces. So, uh, so it's spread through tissue cysts. That's why that undercooked food is an issue. So if you're not cooking your food properly, those cysts can remain, and what's called oocytes are still present inside of it and live. And if you consume those, you can then become infected very easily. Lastly, this is the origin of the crazy cat lady syndrome. You see in like The Simpsons I have here. You know, growing up, I watched The Simpsons, and I always saw the crazy cat lady and thought it was funny. Actually, she's based off of this exact parasitic disease, and that's the running joke with crazy cat ladies. You know, it's actually a disease, and it's possible that those crazy cat ladies that you know are actually infected with this disease. This is a quick slide on the I guess the chronology of sea otter infection. So you have the Toxoplasma oocyte, and this is a cycle, by the way, so it doesn't start in any particular place. But so you have the oocyte infecting these different warm-blooded creatures, mostly these feral animals. It can infect wild felines, but it's mostly the domestic animals that are the issue, especially feral cats that people are taking care of, but they allow them to run around and do their own thing at night. Those are the biggest issue. So they become infected. They then poop, or they poop near a stream. Rain comes down, washes it into the freshwater runoff that goes into the ocean, and then the sea otters become infected. Or the sea otters' prey choice, which one of their main ones is, we'll see later in this, the mussel and these marine snails, which they tend to eat. These are both big issues. All right, so the timeline. So in 1991, it was known that the sea otter population was not bouncing back from their status as an endangered species, but the reason for their high mortality rate was still unknown. Research in 1992 began on sea otter mortality in Monterey Bay specifically, and in 1996, Toxoplasma Gondii was identified as a significant cause of the sea otter mortality in that population. Research continued, but shifted more towards parasitology, knowing that that's now the issue. They shifted gears a little bit, and they wanted to study the origins and that cycle that we just went over. The geographic distribution and the chronology of the development of this protist. Its timeline, basically, its life. In 1998, given that the disease could infect humans, there was a big boom in research and interest in this disease. And scientists from many different backgrounds came together to study the disease. In 2002, Melissa Miller of the California Department of Fish and Wildlife established the first link between sea otter infection and freshwater runoff. So like I was talking about, it tends to come downstream into the marine environment that these sea otters are living in. She was the first to establish that link. Serological testing was done, and now we actually still use the same testing that she used in order to test these sea otters for the disease. We still use that same test to test marine mammals for this disease. In 2004, it was realized that sea otters were being infected by a unique strain of the disease. So we knew that it was Toxoplasma Gondii, but this specific strain tended not to show up a lot in nature. So the major break was finding the DNA link between the sea otter parasites and the feline feces. So we finally found that link, and that showed to be a very significant contributor to these sea otters being infected. And this was found not directly on the beach or near them, but actually way upstream, which was interesting. So in 2005, after these links had been established, the NSF, National Science Foundation, awarded a $1.86 million grant to collaborators from many different California institutes. So in 2006, the researchers honed in on the poo. So now they were attacking that specific point and trying to figure out more about it. So upon further investigation, like I said before, the disease didn't seem to be coming from these wild felines in the area, like the bobcats and wild cats. It looked like it was coming more from these domesticated animals, like people letting their cats out at night, like I was talking about. That became more clearly a significant issue for these sea otters than the wild animals of the area. So in 2009, Dr. Christine Johnson narrowed down the risk factors, which was a big help for tackling this disease. She found that behavior and prey choice significantly impacted the infection likelihood. And by that I mean, so if a sea otter has a choice, and this particular population of sea otters really likes to eat the marine snails in the area, they had a much higher chance, a much higher risk of infection than some other sea otters in the area because of the marine snails. They just had a higher risk of infection than, say, the mussels in the area. So in 2010, due to the success of the first grant, another grant was given, again to a team of collaborators for $2.5 million. So ongoing, 2015 and up, the research is still being conducted with more emphasis on the food cycle and the pathology and parasitology. In conclusion, studies are still ongoing. Sea otters have seen small shifts in population size, both good and bad. So although we've identified this since the 2010 era, we haven't really done much about it. From what I could find in my research, we haven't really gone after these, contributing factors like the domesticated animals and stuff. I'm sure things have been done locally, small things like, telling people to keep their cats indoors and whatnot. But, the big picture, we haven't seen a huge shift for the good in the sea otter population in California. So more needs to be done about that for sure. I was expecting to see a big change at the end of my research and be able to say, hey, this was great. We identified something like what happens a lot in science, and we attacked that, and it became a really good example of what science can do for species, but I can't say that just yet. So more needs to be done about it. And my references. Thank you, Professor.